Hey everybody, it is your life science / biology teacher, Mr. Poser, and today we are finishing our 7th unit on heredity in this class by discussing Mendelian genetics. And this is where we get into the real probability and Punnett squares and that kind of stuff that you might have heard of before, maybe from a previous science class. So like always, I like to start with a question that we'll hopefully figure out an answer to by the end of the video, or we'll come back to once we have a better understanding of this content here. So I have a question. Do you like broccoli? Do you like Brussels sprouts? Do you like cabbage? Do you like things like kale? Any of that? Do your siblings like all these veggies? Do your parents like all these veggies? And have you ever wondered why some people like them and some people don't? Or like just taste preference in general? Maybe genetics has an answer for that, and that's what we'll talk about towards the end of this video. All right, but let's talk about Mendelian genetics and how traits are passed down. We've spent this whole entire unit talking about how traits are passed down and how genes determine or make proteins, and proteins determine your traits and all that kind of stuff, right? And where are genes located? They're in chromosomes. How do you inherit chromosomes? But here is where we get into the real probability and that kind of stuff when we talk about Mendelian genetics. And the reason why we call it Mendelian genetics is named after this guy, Gregor Mendel. He was a monk. I think he was a monk. Yeah, a monk in an abbey. And since he had a lot of free time, he gardened a lot, and he planted pea plants, and then he did experimental crosses with them, genetic crosses. He controlled the mating between different pea plants by cross-pollinating them to determine which traits were passed on and in what amounts or in what ratios. Okay, so Mendelian genetics is named after these crosses that he did with pea plants, and they're a really, really good place to start when we start to talk about how traits are inherited, and at what percentage or what probability and what ratio can we expect to genotypes and phenotypes, which we'll talk about in a second here. But he derived this theory of how traits are transmitted by studying these kinds of characteristics and traits in pea plants, whether or not seeds were round or wrinkled, maybe they're yellow or green, maybe the flowers were white or purple, violet, red. All sorts of stuff that he used to figure out how these traits are passed down. Okay, and we're going to walk through one of his experiments. It says right here, Mendel observed traits in several pea plants by conducting a genetic cross, or a mating of two organisms, right? And where we start here, one of his most famous experiments here was he crossed a purple flowered pea plant with a white flowered pea plant, and both of them were true breeding, meaning that this purple flower plant only has ever bred with other purple flowered plants, and this white flowered plant has only ever been bred with other white flowered plants. Okay, so they were what we would call purebred. So think like horses and that kind of stuff too. But in this case, we have pea plants. Hey, and this is what we would call the P generation, or the parental generation. Okay, and when Mendel crossed these two plants, all of their offspring, 100% of their offspring in what we call the F1 generation, their kids were purple. None of them were white. None of them had white flowers. They were all purple. So he was wondering, okay, why are they all purple? So then what he did after that, he crossed two plants from the F1 generation and got the F2 generation and found more plants that had this white flower characteristic. Okay? It was weird because the white flower characteristic kind of skipped a generation, or he found that the white flower trait had been masked. Okay, and in this F2 generation, it's called, when we cross two individuals from this generation to the next generation, the offspring demonstrated a 3-to-1 purple flower to white flower ratio. That means 75% of the pea plants that he crossed were purple, only 25% of them had these white flowers, right? So Mendel, is just figuring this out, like based off of, I'll cross these together and then I'll count how many are purple, how many have white flowers, so on and so forth. He didn't know about genes. He didn't know what genes were. He didn't know about meiosis. He didn't really even know about DNA, okay? But he had to figure out that there was some kind of sort of heredity factor that stored information that allowed these plants to express their genes. There was something getting passed down from one generation to the next. And as we've talked about ad nauseum in this class, it's DNA, it's genes, right? And that's what we're really going to look at here by studying Punnett squares in a little bit here. But genes as we should know, are passed through generations, right? So that white flower gene was passed down, and it wasn't shown in that F1 generation, but it was shown in the F2 generation. And genes are responsible for our traits. I've said that a bunch of times if you've been paying attention in this class, right? Each gene has what we call a locus, and in fact, I'm going to highlight that because that's going to be a good term to know. A locus, which is location on a pair of homologous chromosomes, right? So this is what this is human chromosome number 20, and these are just a few of the genes that are located on chromosome number 20, and they kind of appear as like stripes on the gene. So what we've been looking at before, when we were studying karyotypes earlier in this unit, we saw that some of these chromosomes had stripes on them, and those are different genes. Okay, and there's bunches of genes located on chromosomes, but these are some of the major ones that we might find on human chromosome number 20. And don't ask me what these stand for, because I really don't know. Maybe at one time in my life I knew what these stand for, but these are all different genes located on a human chromosome, right? So chromosomes contain the genes, and the genes are passed down. Genes determine your traits. We've been over this before, okay? But what made these flowers different from one another is they had both a gene for flower color. They both had genes for flower color, right? But they had different versions of the gene for flower color, okay? And those versions of those genes are what we call alleles. And those are alternative forms of genes that may occur at a specific locus, right? So a gene is going to be located on a chromosome at what we call a locus, but you might have the recessive gene, or you might have the dominant gene. And maybe you've heard those words before in another class. Hopefully they're ringing some bells a little bit, okay? But since we are diploid organisms, and so are those pea plants, diploid cells have two alleles for a gene. They have two copies of the same gene, okay? Two copies of the same gene because they are diploid, and that's what we've been talking about when we were discussing meiosis and chromosomes. Diploid means you have two copies, two sets of chromosomes, right? So because you're diploid, you have two copies of every gene, and you might have two of the same version of the gene, or you might have two different versions of the gene. Then that might mess with your traits a little bit, okay? So if you have two of the same allele, if you have two of the same gene at a locus, let's just say this is a homologous pair here, and you have, or let's just say, we'll put this back into pea plants here, okay? Let's say a pea plant has this pair of homologous chromosomes, and take a look. They have the same gene for at the same locus for purple flower color. They are what we call homozygous. They have two of the same allele at a specific locus. And if they have one copy of the purple flower gene, or the purple flower allele, and one of the white flower allele, then they are heterozygous. They have two different alleles at the same locus. Okay, so like I said, every single diploid organism, every single diploid cell has two copies of that gene, right? But whether they are homozygous or heterozygous determines, what trait they're going to express, or what trait or they're not going to express. Okay, so are these flowers here, let's just say this is one flower, they have two alleles for the purple flower, and this only has one allele for the purple flower, one allele for the white flower. Is the pea plant going to be purple or white? I guess we'll find out here in just a second. A dominant allele is expressed even when two different alleles are present. Okay, so the answer to the question that I just posed before is that both of these flowers, or both of these pea plants, if they have these two alleles, that means they're both going to be purple, which is the dominant trait. They're both going to have purple flowers. If you have one copy of the purple flower allele, or if you have two copies of the purple flower allele, you're going to have purple flowers if you're a pea plant. Okay, even if you have one white flower allele, you're still going to be purple. Okay, why is that? Well, purple is dominant and white is recessive. The white flower allele is recessive. So a recessive allele is only expressed when two recessive copies occur together. Only if you get the recessive allele from mom and the recessive allele from dad are you going to have white flower colors if you're a pea plant. Okay, and what we call is an organism's genotype is its genetic makeup, or the pairs of alleles that it has. Check it out. This pea plant over here, we have two alleles for white flowers, so which means that this plant is going to have white flowers, unlike its purple brothers and sisters. An organism's phenotype is physical characteristics, which is determined by its genotype. Now, I'm going to need you to remember those two terms and not switch them up. Genotype are the pairs of alleles or the genetic makeup of an organism. A phenotype is the actual traits that is expressed. Okay, so a phenotype in this case, you either have purple flowers or you have white flowers. A genotype, well, it could be three different things here. Let's take a look in a Punnett square. Did I skip a page? Nope, I did not. Okay, alleles and genotypes are represented by letters, and we can use a Punnett square to predict results of crosses just like Mendel did using these letters here. Okay, so if you see two capital letters paired next to each other, that's what's called a genotype. Okay, and if you got two capital letters, or each pair of letters is called a genotype. Let me rephrase that. Each pair of letters is a genotype. If you've got two pairs of capital letters, those are representing two dominant alleles. And the word for that is that they are homozygous dominant, two dominant alleles. Okay, homo means same, hetero means different, right? So if you have two different copies of the gene, if you have two different versions of the gene, then it means that it is heterozygous. Okay, hetero means different. And then two lowercase letters. Okay, so an uppercase and a lowercase letter means you're heterozygote. Two lowercase letters means this organism is homozygous recessive. Two copies of the recessive allele. Okay, so let's cross these two flowers in our Punnett square and let's see what happens. Let's model out Mendel's first cross here. Okay. So this is the true breeding P generation. If we put these together on a Punnett square, what we can do, we put the parents' genotypes are written outside of the square like this. So this is one of the parents' P plants. It is homozygous dominant. And here's the other parent, the white-flowered parent, and they are homozygous recessive. If we put them on a Punnett square, all we have to do, really, the next step is cross those genotypes. Okay, so we're going to bring this A down to this box and this box, bring that A down to this box and this box, use that column, and then we'll bring over each of the recessive A's into the two rows. And then what we'll get from that are four different genotypes, four different pairs of alleles. Okay, so it says the genotypes are crossed and the results will predict the genotypes of the next generation. So a complete Punnett square, if you want to try this out yourself, go for it. You can stop me right here, but I'm going to switch to the next slide. The complete Punnett square will look like this. And once again, the capital A represents the dominant allele, and the recessive A represents the lowercase a represents the recessive allele. So what color are all these flowers going to be based on the genotypes that we're seeing in these boxes? Okay, what color are they going to be? If you already know, great. If you don't, pause and try to answer it. But they will be purple because they are all heterozygous and they have one copy of this dominant allele, and that's all they need in order to express the dominant phenotype in order to be actually purple. Okay, so all the flowers are purple, just like Mendel had when he crossed this true breeding purple flowered pea plant and the true breeding white flowered pea plant. Okay, so if we cross the F1 generation, take a look. We have two heterozygotes here just from our last cross. So we take two of these flowers here and we cross-pollinate them. What's the result going to be? Here's our big A, little a, big A, little a. If we do this Punnett square, okay, what's gonna come out from these F1 generation here? So work this out. Try to work this out. Pause the video if you have not already, and see if you can't make this Punnett square in your notes. Okay, but here they are. We got big A from this cross here, big A, little a from this cross, big A, little a in this cross, and little a, little a. So if you got that far, great. Okay, but then it's figuring out what they mean, like deciphering those genotypes here. Okay, so what are the phenotypes that each of these four pea plants are going to express? Okay, well, let's find out. This one's going to be purple. It's got two dominant alleles. This one's going to be purple. It's got a dominant allele. This one's going to be purple. It's a heterozygote. It has one dominant allele. And then look at our guy over here. Got two recessive alleles, and thus is going to bring back the white flowered phenotype. All right, so we can expect 75% or 3-to-1 purple flowers to white flowers. Only our white flowers are going to be about 25% of our offspring if we cross these two plants. Okay, and this is the 3-to-1 ratio that Mendel found in his experiments. Okay, so back to the Brussels sprouts and cabbage and all that stuff. Maybe you do, maybe you don't like them, maybe your brothers and sisters do. Okay, but if we go into this here, maybe there is a genetic basis. Broccoli, cabbage, and Brussels sprouts contain a chemical called PTC, or phenylthiocarbamide, which is a bitter chemical that can be only tasted by people with a homozygous dominant or heterozygous genotype for a particular gene. Okay, so just like with the pea plants here, this is a trait that's, or what we would call a Mendelian trait in people. Right? So if you have 1 or 2 copies of what's called the PTC tasting gene, then if you put this piece of paper on your tongue, see, check it out, it is PTC test paper. Okay, if you take this and put it on your tongue, then you're going to taste a very bitter chemical that's often found in these types of vegetables. Guess what? I am not a I am not a taster. That means I have 2 recessive alleles, and I cannot take a PTC. So that means that my genotype here is going to be little t, and I am homozygous recessive for PTC tasting. 70% of people can taste it. I guess I'm in the 30% here that cannot taste it, or 70 to 75% can taste it. All right, but let's get a little bit more into Mendelian genetics here. Let's continue this on. 1 in 12 men, actually, you know what? Let's save this for another video. Let me know if you have any questions. We'll see you next time. Bye.